Chapter 3, Plant Kingdom, by Dr. Jayachandran Tejas. Earlier, Monera, Protista, fungi and blue-green algae were considered as part of plant kingdom. But presently, they are not. Blue-green algae are actually not true algae, they are cyanobacteria. Plant kingdom is divided into algae, bryophytes, pteridophytes, gymnosperms and angiosperms. Angiosperm classification. Angiosperms refer to flowering plants. There are two classification systems to classify angiosperms: artificial classification system used by Linnaeus and natural classification system used by George Bentham and Joseph Dalton Hooker. In the artificial classification system, equal weightage was given to vegetative and sexual features of plants. Vegetative features are habitat, color and leaves. Sexual features refer to androecium structure. This led to separation of closely related plants as vegetative features are easily affected by the environment. Natural classification system was based on natural affinities between organisms. This used both external features and internal features. Internal features include ultrastructure, anatomy, embryology and phytochemistry. Phylogenetic classification system. Phylogenetic classification system is based on evolutionary relationships between organisms. Organisms belonging to same taxon have a common ancestor. Fossils are mainly used for this classification system. Numerical taxonomy. This method of taxonomy uses computers. All characters of the organism are used, thus hundreds of characters can be used. Each character is given a number or code. All characters are given equal importance. Cytotaxonomy uses cytological information like chromosome number, chromosome structure and chromosome behavior. Chemotaxonomy used chemicals found in plants. Algae. The main feature of algae is their thalloid structure. Thalloid means no organization into roots, stems or leaves. Algae are mostly aquatic. Some are found on moist rock, moist soil and wood. Some are found in association with other organisms. Examples are lichen, which is a combination of algae and fungus, and algae found on the body of sloths. Algae are found in various forms. Eulothrix and Spirogyra are filamentous algae. Volvox has colonial form. Kelps are massive algae. Algal reproduction may be vegetative, asexual or sexual. Vegetative reproduction is by fragmentation. Asexual reproduction is by spores, usually zoospores with flagella. Sexual reproduction is by fusion of gametes. Gametes may be of same size isogamous or of different sizes anisogamous. Examples of isogamous gametes are Eulothrix, flagellated gametes, and Spirogyra, non-flagellated gametes. Example of anisogamous gametes is Eudorina. Oogamy is a special type of anisogamy. Here, a smaller mobile male gamete fuses with a larger non-mobile female gamete. This is seen in Volvox and Fucus. Algae are very useful. Half of the carbon dioxide fixation in the world is by algae, thus they produce large amount of oxygen. Algae are the primary producers in aquatic food cycles. Some algae are used as food. About 70 species of marine algae are used as food. Examples include Porphyria, Laminaria and Sargassum. Chlorella is a unicellular alga which is used as a protein supplement by space travelers. Algae can be used to make hydrocolloids, water-holding substances. Examples are algan from brown algae, carrageen from red algae and agar from Gelidum and Gracilaria. Agar is used to grow microbes and to prepare ice creams and jellies. There are three types of algae: green algae or Chlorophyceae, brown algae or Phaeophyceae and red algae or Rhodophyceae. Green algae are found in fresh water. Brown algae and red algae are found in marine water. Red algae are found in warm water, both near the surface and at depths. All algae have chlorophyll in chloroplasts. Green algae have chlorophyll A and B. Brown algae have chlorophyll A and C and red algae have chlorophyll A and D. Brown algae also have fucoxanthin or xanthophyll, which gives brown color, and carotenoids. Red algae have R-phycoerythrin, which gives red color. Green algae store food as starch. Storage bodies are called pyrenoids. These are present inside chloroplasts, and contain both starch and protein. Brown algae store food in the form of two complex carbohydrates, mannitol and laminarin. Red algae store food as floridine starch, which has a structure similar to amylopectin and glycogen. One peculiarity of the cell structure of brown algae is the presence of a central vacuole. Cell wall in green algae is rigid and is made up of cellulose inner layer and pectose outer layer. Cell wall in brown algae has cellulose inner layer and algin outer layer. The algin outer layer gives a gelatinous appearance. Cell wall in red algae is made of cellulose, pectin and polysulfate esters. As earlier told, asexual reproduction is by spores. Green and brown algae have zoospores, that is, with flagellae. Red algal spores do not have flagellae. Green algae have 2 to 8 equally long, apical flagellae. Brown algae have 2 unequal lateral flagellae. Spore of brown algae is pear-shaped. Examples of green algae are Volvox, Eulothrix, Spirogyra, Chlamydomonas and Chara. Examples of brown algae are Laminaria, kelp, which is profusely branched and may reach 100 meters, Fucus, Dicteata, Ectocarpus, made of simple branched filaments, and Sargassum. Examples of red algae include Porphyra, Polysiphonia, Gracilaria and Gelidium. Structure of brown algae needs special mention. It has 3 parts: holdfast, stripe and frond. Holdfast is the part which attaches to substratum. Stripe is the stalk. Frond is the leaf-like photosynthetic organ. Bryophytes. Bryophytes include mosses and liverworts. They are seen in damp, humid shaded areas like side of hills. They are called amphibians of the plant kingdom as they live on land and need water for sexual reproduction. Bryophyte structure includes a thallus-like body and root-like structures called rhizoids. The thallus-like body may be erect or prostrate. This thallus is more differentiated than that of algae. Still, there are no true root or stem or leaves. Rhizoids attach the thallus-like body to the substratum. Rhizoids may be unicellular or multicellular. The plant body of a bryophyte is haploid and is called gametophyte. It develops male sexual organ called antheridium and female sexual organ called archegonium. Antheridium releases biflagellate male gametes called antherozoids into water. The flask-shaped archegonium has a single egg. Antherozoid fuses with the egg to form a zygote which is diploid. The zygote does not undergo reduction division, meiosis. Instead, it grows to form a multicellular body called sporophyte. Sporophyte is attached to the gametophyte and receives nutrition from it. Sporophyte has 3 parts: foot, seta and capsule. Sporophytes of mosses are more elaborate than that of liverworts. Some cells of the sporophyte undergo reduction division, meiosis, to form haploid spores which germinate elsewhere to form a new gametophyte. Bryophytes are important in many ways. Some mosses are used as food by animals. Sphagnum, a moss, provides peat, which is used as a fuel and also is used as packing material for transshipment of living material, as it can hold water. Mosses and lichens were the first organisms to colonize rocks. They decomposed rocks, making growth of higher plants possible. Mosses form dense mats on the soil. This reduces the impact of rain and prevents soil erosion. In liverworts, the gametophyte has a dorsiventral thallus that is closely oppressed to the substrate. Leafy members have tiny leaf-like appendages in 2 rows on stem-like structures. Asexual reproduction is by fragmentation and by formation of buds called gemmae. These develop on gemmacups on the thallus and then detach to form new plants. Sexual reproduction is by the archegonia and antheridia on the thallus. An example of liverworts is Marchantia. In mosses, the gametophyte has 2 stages. The first stage, which develops from the spore, is made of creeping green branched filaments called protonema stage. From this, leafy stage develops upright slender axis bearing spirally arranged leaves and attached to the soil by rhizoids. Asexual reproduction is by fragmentation and by budding from secondary protonema. Sexual reproduction is by archegonia and antheridia which develop on the apex of the leafy stage. Examples of mosses are Funaria, Polytrichum and Sphagnum. Pteridophytes. Pteridophytes include ferns and horsetails. They are the first terrestrial plants to possess xylem and phloem. They are usually found in cool, damp, shady places. They are used as soil binders, for medicinal purposes and for ornamental purposes. The main plant body is a sporophyte, not a gametophyte as in bryophytes. It has true stem, leaves and roots. Also, it has vascular tissues, xylem and phloem. The leaves may be small, microphyls as in Selaginella, or large, macrophyls as in ferns. Some leaves have reproductive organs called sporangia. Such leaves are called sporophylls. Sometimes, sporophylls develop strobily compact corn-like structures seen in Selaginella and Equisetum. Sporophylls are leaves which subtend sporangia. Inside the sporangia, spores are formed by meiosis. These spores germinate elsewhere into a gametophyte called prothallus, which is thalloid photosynthetic. This needs cool, damp, shady place to grow, so pteridophytes are restricted to narrow geographical regions. The gametophyte bears antheridia and archegonia. Antherozoid from antheridia are carried by water to egg in archegonium, where it fuses with the egg to form a zygote. The zygote develops into a sporophyte. As just told, sporophyte releases spores. Usually these spores are all the same, called homosporous. Sometimes, the spores are different heterosporous. Examples are Selaginella and Salvinia. The two types of spores are microspores and macrospores. Microspore germinates to form a male gametophyte and macrospore germinates to form a female gametophyte. Female gametophyte is retained on the sporophyte for a variable time. This event is a precursor to the seed habit, the next step in evolution. Pteridophytes have four classes: Silipsida, Lycopsida, Sphenopsida and Pteropsida. Gymnosperms. Gymnosperm means naked seed, meaning that seeds are not covered by fruits. This happens because ovules are not enclosed by ovary, and so seeds remain exposed. Gymnosperms include trees and shrubs. The giant redwood tree sequoia is a gymnosperm. Gymnosperms have taproot system. Pinus has fungi in its roots, in the form of mycorrhiza. Cycas has coralloid roots having nitrogen-fixing cyanobacteria. Leaves of gymnosperms are adapted to extremes of weather. Conifers have needle-like leaves. This is an adaptation to reduce surface area to reduce water loss. These leaves also have thick cuticle and sunken stomata for the same purpose. Cycas has pinnate leaves which last for few years. Some gymnosperms, like Cycas, have unbranched stem. Others, like Pinus and Cedrus, have branched stem. Gymnosperms are heterosporous, that is, they produce microspores and megaspores. Spores are formed by reduction division and thus are haploid. Male spore develops into a male gametophyte. Female spore develops into a female gametophyte. Male gametophyte produces a male gamete and female gametophyte produces a female gamete. Male gamete fuses with the female gamete. This is called fertilization. This fused cell is called the zygote. The zygote develops into an embryo. This embryo is enclosed inside the seed. Seed is formed from ovule. Seed is not covered by fruit. The seed germinates into a new gymnosperm plant. Spores form on structures called sporophylls. Sporophylls are arranged spirally along an axis to form strobili or cones, which may be compact or lax. Sporophylls have sporangia which produce spores. There are male strobili and female strobili. Pinus has male and female strobili on the same plant. Cycas has male and female strobili on different plants. Male strobili have microsporophylls bearing microsporangia. In the microsporangium, microspores develop into multicellular male gametocytes called pollen grains. Pollen grains are released into the wind. Female strobili have megasporophylls bearing megasporangia. Inside the megasporangium, there is a structure called nacellus, which is protected inside an ovule. One cell of the nacellus becomes the megaspore mother cell. This undergoes meiosis to form 4 megaspores. One megaspore develops into the multicellular female gametophyte. This female gametocyte has 2 or more archegonia, female sex organs. Each archegonium has an ovum. Pollen grain is carried by air currents. The pollen which comes into contact with the opening of ovule will generate a pollen tube towards the archegonia. Pollen tube has male gametes. A male gamete will fuse with an ovum to form the zygote. Note that, unlike in bryophytes and pteridophytes, in gymnosperms male and female gametophytes do not have independent existence. Angiosperms. Angiosperms have flowers. These are specialized structures having pollen grains and ovules. Ovules are inside ovary and so seeds are inside fruits. Angiosperms range in size from small wolffia to large eucalyptus tree. Angiosperms are divided into 2 classes, dicotyledons and monocotyledons.